Hello friends, today I have a new set of slides which is very interesting, entitled The Story of Genes. So let us go through. The topics under discussion include what is DNA, what are nucleotides, speaking about the composition of DNA, structure and composition of DNA next, Chargaff's rule, DNA versus chromatin versus chromosome. Then I'll speak about DNA packaging in eukaryotes, DNA packaging in prokaryotes. Then genes versus alleles, DNA inventions, functions of DNA, rules of DNA, locations of DNA in prokaryotes and eukaryotes, physico-chemical properties of DNA, COT, that is the COT curve and COT value. What does it indicate? We'll speak about that. Recap, a short recap. Now coming to what is DNA? DNA, the full form, as we all know, deoxyribonucleic acid is a macromolecule. At the end of the DNA isolation experiment, we are able to see the DNA in actual. So it's a macromolecule made up of nucleotides responsible for carrying the genetic instructions from one generation to another. You know very well that the genes responsible for carrying the genetic material from one generation to another generation. Genes are nothing but segments of DNA, and they code for a particular amino acid, precursor of protein. So DNA, deoxyribonucleic acid. Now you may ask me, what are nucleotides? We said no, DNAs are made up of nucleotides. So what are these nucleotides? Nucleotides in turn can be broken down to nucleosides with S and phosphates. Nucleosides are made up of pentose sugar and nitrogenous bases. So pentose sugar can be, in the case of DNA, it is deoxyribose in case of DNA, and in case of RNA, it is ribose sugar. I repeat, in case of DNA, it is deoxyribose, that is the pentose sugar present in DNA, and in case of RNA, it is ribose sugar. That's about the pentose sugar. Nucleosides are made up of nitrogen bases also. That includes adenine, guanine, cytosine, and thymine. Adenine and guanine are the purines, that is the fused ring structure. Double ring structure is a fused ring structure. Purines and cytosine and thymines are made up of a single ring structure. They are pyrimidines. So nitrogenous bases are composed of adenine, guanine, cytosine, thymine. Next, going on to the types of DNA. There are mainly three types of DNA, that is A form of DNA, B form of DNA, and Z form of DNA, out of which B form of DNA is the most common one. A form of DNA, as you can see, is the right-handed double helix. B form of DNA is also right-handed double helix structure. That twisted ladder form is called the double helix structure. That is the B form of DNA, most common. And Z form of DNA is the left-handed double helix structure. It's also a double helix structure, but left-handed double helix structure. That is the Z form. And you can see in between there are some curly images of the DNA strand. That is the characteristic of the Z form. Speaking about the structure and composition of DNA, as we all know, the twisted ladder structure of the DNA is known as the DNA double helix. This structure is known as the DNA double helix structure. We all know that DNA, A for DNA means acid. It's a nucleic acid, deoxyribonucleic acid. Name roots from deoxyribose sugar. Nucleic acids can be broken down to nucleotides. We have just seen it. And nucleotides are made up of nothing but pentose sugar, phosphate, and nitrogenous bases. Nitrogenous bases can be adenine, guanine, cytosine, and thymine. Out of which, in adenine and thymine, there are a double bond, double hydrogen bond, where I am pointing out. Please see it carefully. There are two bonds of hydrogen. That is, double hydrogen bond is present between this A representing adenine and T representing thymine. So there are two hydrogen bonds. And in between C, cytosine, and guanine, there are three hydrogen bonds. Remember this. In between A and T, there are two hydrogen bonds. And in between C and G, there are three hydrogen bonds. The sequence, that is A, T, G, C, not necessarily that in every organism A will occur after T, T will occur after G, G will occur after C, not necessarily. It can occur in any order. It can be TA, CG or TGCA, CGTA, any form, any order. That's why we all are unique. We all have individual characteristics because of the sequence of DNA, unique sequences within the DNA. That's why DNA varies from individual to individual. Hence, the genes vary from person to person. Now, let's go on to the structure and composition of DNA, speaking about the bonds in the DNA. Okay, now we all know that thymine, T, that is the purine, that is the pyrimidine, that is the nitrogenous base. Thymine is the nitrogenous base that clumps with or connects with pentose sugar and phosphate to form nucleotide. Now first let us speak about this linkage. This linkage, you see an N and link to pentose sugar. Let's speak about this linkage. You can see very well that nitrogenous base, that is N1 in pyrimidine. Pyrimidine is nothing but here the thymine. That's the pyrimidine over here. That pyrimidine, N1 of pyrimidine, this is N1 of pyrimidine, is linked with the first carbon of the pentose sugar. Let us see it. This is the pentose sugar, and the first carbon of the pentose sugar, this first carbon of the pentose sugar is linked to N1 of pyrimidine, that is thymine. I'm talking about this linkage, and this linkage is known as the N-glycosidic linkage. As this due to this N-glycosidic linkage, as pentose sugar is linked with the pyrimidine, that is thymine, nitrogenous base, we call it a nucleoside. You know very well, sugar plus nitrogenous base equal to nucleoside. For making a nucleotide, you need an additional phosphate group, isn't it? Let us see how the bonds are made. We need an additional, this is the phosphate group. We need a phosphate group which should be linked to this nucleoside to form nucleotide. So this T group joins to the 5' OH of the nucleoside, meaning that, see, this pentose sugar, it's linked to the one of the carbon of the pentose sugar. This is 1, this is 2, this is 3, this is 4, and this is the 5th carbon. That is, the OH on the 5th carbon is linked with the phosphate group. This is the 5th carbon of the pentose sugar which is linked to the phosphate group to form ultimately a nucleotide. This 3, where I am encircling my pointer, this is a nucleotide, a complete nucleotide. Now, this nucleotide must be linked with sidewise to another purine, that is the adenine. So, how it links? Through hydrogen bonding. As I told you, P-thymine. Between P-thymine and A-adenine, there are 2 hydrogen bonds. See clearly, there are 2 hydrogen bonds. And since adenine is a purine, it is having a fused ring structure. So now, this is again linked to the pentose sugar. How? N9 in purine, adenine, is linked to the pentose sugar. This is the N-glycosidic linkage, as you can see it. Let's number it: 1, 2, 3, 4, 5, 6, 7, 8, 9. This is the 9th numbered nitrogen which is linked to the first carbon of the pentose sugar. See here, N9 in purine is linked to first carbon of the pentose sugar. So I am talking about this as the N-glycosidic linkage. This as the N-glycosidic linkage. So here also nitrogenous base plus pentose sugar is equal to nucleoside. You know very well now, nucleoside must be linked with the phosphate group. So another phosphate group is also linked to it. Same as here, the phosphate group is linked to the nucleoside over here. How the phosphate group is linked? See here, 1, 2, 3, 4, and 5. The 5th carbon, the OH of the 5th carbon is linked to the phosphate group over here to form ultimately the nucleotide. This whole square region is nothing but the nucleotide containing the pyrimidine, that is the purine, that is the adenine, sugar, and phosphate. Another case, this is one way about A and T. Say about A and T over here. Now it has to lengthen, right? The DNA is a long stretch of nucleotides. So it has to lengthen. How will it lengthen? Through this bond. See, 1, 2, 3. The 3rd carbon of the pentose sugar to this, the phosphate group, links. And it connects to the 5th carbon of the next pentose sugar. That's why the backbone is named as 3', 5' backbone. Sugar, this is the pentose sugar. Let's number it: 1, 2, 3, 4, and this is the 5th carbon. That is the 5' OH on the 5th carbon is linked to the 3rd carbon of the previous pentose sugar. This is 3' and this is 5'. In between, we have the phosphodiester linkage. See, this is the phospho 3', 5', phosphodiester linkage. Let's go through this. Pentose sugar from 3' carbon, that is this carbon, 3' carbon, this is the 1, 2 and 3' carbon from one nucleotide, this is one nucleotide, this is one nucleotide from 3' carbon from one nucleotide is linked to the P group bonded with 5' carbon from the next nucleotide. This 3 are the next nucleotide, the nucleotide below cytosine and guanine. Cytosine, pentose sugar and phosphate forms the next nucleotide. You can see the 3' to the 5' bond, that is phosphodiester bond in between this. And then in the same way, cytosine bonds with guanine. In between there are 3 hydrogen bonds. And you know very well, guanine being a purine, it has a fused ring structure. And you can see a pentose sugar in an inverted fashion. The linkage is the N-glycosidic linkage. Then it has a phosphodiester linkage also here. And the story goes on. Like this, the 3', 5' phosphodiester bond links from one nucleotide to another nucleotide to extend the DNA. Okay. So these two are dinucleotides. Again, we can call these two are dinucleotides. Several dinucleotides or several nucleotides form the polynucleotide or macromolecule. Hence, this macromolecule is termed as DNA. I hope the bondages are clear. These are the bonds, very important bonds, glycosidic bonds, phosphodiester bonds, which must be remembered. Next, this is a summary of the structure and composition, which is very important from exam point of view. Nitrogenous bases, you know very well, adenine, guanine, cytosine, thymine, out of which adenine and guanine are the purine, cytosine and thymine are the pyrimidine. You know very well, cytosine and thymine are having the single ring. Here, see the structure, single ring, whereas adenine and guanine, that is the purine, are having the fused ring structure. Here, fused ring structure. Remember this. So, pentose sugar, nitrogenous base, phosphate, and this sky color is the backbone, made up of 3' to 5' phosphodiester bond linked with pentose sugar. Sugar, we call it sugar phosphate bond. 3 parts of nucleotide. Thus, we have nitrogenous base, sugar, and phosphate. See, it's linked to 1' carbon. This third carbon links to the next nucleotide, and this 5th carbon links to the phosphate group. So this is about the structure and composition of DNA. Next, going on to the Chargaff's rule. Chargaff's rule says that A, adenine, a purine, should be equal to and must be equal to thymine, that is a pyrimidine. A equal to T, and G, another purine, must be equal to C, that is cytosine. And the total of A plus G, that is adenine plus guanine, that's the amount of purine, must be equal to the amount of pyrimidine, that is purine equal to pyrimidine. This law is stated by Chargaff and is known as the Chargaff's rule. The DNA is made according to the Chargaff's rule. Then coming on to DNA versus chromatin versus chromosome. These are very important terminologies as far as genetics is concerned. Please listen to this carefully. We know a cell, a eukaryotic cell, that is having a nucleus. Within the nucleus, the chromosomes are present. The story of genes says that it is made up of genes are a segment of DNA. And I'm telling you now that the genes, that is DNA, are packed within the chromosome. These are the chromosomes. Imagine a sweater. You cut the ends of the sweater, then threads will come out, right? If you imagine in the same way that the sweater is made up of the woolen threads, same way the chromosome, here the chromosome is made up of the DNA, and around the DNA and in the center of the DNA is coiled around the histone proteins. Histone proteins. Understood? See the chromosome magnified as this chromosome. Chromosome is made up of DNA, and this DNA is packed around the histones, octamer histones. And this is the double helix structure of DNA, the pre-DNA. Let's get more clarity on this. DNA packaging. How is DNA packaged into eukaryotes? In the same way, we will learn more about this chromatin, chromosomes, and DNA. See, first let us look here. You have seen the nucleus. Within the nucleus, the chromosomes are packed. These chromosomes are packed with what? The DNAs. DNA is a long sequence. It is packed with the DNA, just like the sweater is made up of the woolen threads. So its woolen threads are these ones, which can be magnified and stretched like this. If you magnify a portion of this chromosome, you will see that the DNA is packed around the protein, that is the octamer, that is histones. It's packed around the histones. So we all know DNA plus histone gives the nucleosome. Then what is chromatin? See, chromatin is nothing but a number of nucleosomes. A single unit of this chromatin, that is this nucleosome, that is addition of DNA plus histone forms the nucleosome. When number of nucleosomes are there packed together, we call it chromatin. See here, 30 nanometer of chromatin fiber of packed nucleosomes. A number of packed nucleosomes are nothing but chromatin. Beads on a string. It's known as beads on a string. Here there are 3 nucleosomes, which are known as chromatins. And this chromatin again condenses and gets packed into chromosome, as we have seen it down. Again, if you magnify this DNA that is linked to the histone, you'll find a long sequence of DNA double helix as this. That means DNA double helix is coiled around the histone proteins, which is an octamer. Okay, like this. So I hope it is clear. For the chromosome, packed form, a sweater. If you pull the threads of the sweater, the woolen threads will come out like this. In this case, the woolen threads is coiled around the histone and to form the nucleosome, and several nucleosomes are called chromatin. Thereby, the chromatins are packed in the form of chromosome. This is the magnified sequence which is this upper layer of thread around the histone. So about DNA packaging in prokaryotes. Imagine a bacterial cell. It's having no nucleus. So where is the DNA present? It is present in the cytoplasm as a supercoiled structure as this. If you magnify this portion, you will see it is coiled around a non-histone, that is DNA binding protein. And each structure, supercoiled structure, is anchored by some anchoring protein, as you can see here. And the whole of this is dispersed in the cytoplasm as a supercoiled structure within the bacterial cell. So this is about DNA packaging in prokaryotes. Genes versus alleles. What are genes and what are alleles? Genes, as I said, are segments of DNA coding for a particular amino acid that is precursor of protein. So genes are segments on DNA. DNA means they must be present on chromosome. Got me? DNAs are present where? On the chromosome. DNAs are packed tightly in the form of chromosomes, right? So the DNA, a particular segment of gene, must be present on the chromosome. These two are homologous pair of chromosomes. Say, for example, the gene responsible for flower colors is present here. Now the flower may be a purple one, maybe a white one, or a light purple one. Three options may be there. So if the gene is if the outcome is a purple flower, that means this allele is expressed. Allele for purple color, that means this allele is expressed. That's the dominant gene. This allele is expressed. So what is an allele? Alternative forms of genes. That is variants of genes. Genes are made up of alleles. That is, two alleles make up a gene. Okay. So here, for example, allele for purple color, and this is the allele for white flower. If the allele for purple color flower is more dominant, then the resultant flower in the next generation will be purple colored. If the allele for white flower is more dominant than the allele, then the next generation flower will be a white color flower. And if both are dominating each other, both are co-dominant, then it will be a light purple color. Okay. Now it depends which allele is expressed. Accordingly, the next generation outcome will be there. Just remember, genes means the overall. Purple or white is the allele. That is the allele for purple color flower or allele for white color flower. But genes means the color of flower, responsible for color of flower overall. Now, alleles may be homozygous and heterozygous. Say this is the homozygous allele from one person, the first person. This is the homozygous allele from the second person, and this is the heterozygous allele from the third person. So from the first person, the homozygous allele is capital A and capital A. You can see it is identical. Okay, it is identical. That means maybe both, for an example, we take father's side, both from father. Here small a, that is again identical allele. That is identical alleles, that is called homozygous alleles. Say from the mother's side, the person has inherited small a, say responsible for skin color. Again, in case of heterozygous allele means non-identical allele. One from father and one from mother maybe. Now whether father's gene will be expressed or mother's gene will be expressed or both will be co-dominants, that depends upon this allele which will be expressed, which is the dominant or both are dominant. Anyway, this is the homozygous allele, that is identical alleles, as you see, capital A, both from father, for example, small a, both from mother, for example. Identical allele means homozygous allele and heterozygous allele means it is non-identical. That is, capital A may be from father, small a may be from mother. It depends. What will be the skin color, for example? If father's gene is expressed, then it will be somewhat A represents whatever A represents. And if mother's gene is expressed, it will be from whatever small a represents accordingly. So I hope you have understood alleles and genes. Let's go on. DNA inventions. DNA was initially identified by Swiss biologist Johannes Friedrich Miescher in 1869 while working with WBCs, that is white blood cells. Later, the double-helix structure was confirmed by James Watson and Francis Crick. This is very important, James Watson and Francis Crick. Now coming on to functions of DNA. You see a cell, you see chromosomes. From chromosomes, the DNA is being pulled out. This DNA is actually coiled around histones. This is all for your remembrance. Now DNA contains genes, segments of DNA, genes which are responsible for coding onto the amino acids, that is precursor of proteins. We know from DNA we get RNA and from RNA we get proteins. So DNA to RNA is known as transcription. So another function of DNA is transcription. Then replication of DNA must be there. So another function is replication. Then you have cellular metabolism. Whatever gets digested into our body is because of metabolism, that is cellular metabolism. DNA fingerprinting means a particular person is identified based on his nucleotide sequence, unique nucleotide sequence. That's identification of person based on DNA fingerprinting, as we all know. Gene therapy means a missing DNA link or a missing or a damaged DNA is replaced by a healthy DNA to cure a particular disease, gene therapy. So these are the functions of DNA. DNA grooves. DNA is having two grooves, the major groove and the minor groove. The major groove occurs where the backbones are far apart. See, this is the sugar phosphate backbone, this yellow, and this is another sugar phosphate backbone on the other side. In between, a large space is there as compared to the minor groove. A large space is there. So it is known as the major groove. The major groove says that the backbones are far apart. The backbones are far apart. Next, the minor groove says that the backbones are close by, close together. See, there is hardly any space between this portion and this portion. It's closed. So it's hardly any space. That's the minor groove. See here, this is the major groove and this is the minor groove. Okay, so this is about the DNA grooves, remember. Location, as I told you when I was explaining about the packaging of DNA in eukaryotes, the DNA is obviously located in the nucleus in the form of chromosomes. You know, chromosomes from chromatins, and chromatins are made up of nucleosomes, that is DNA plus histones. DNA contains the genes. Again and again, I'm telling this so that it's right into your memory. So in short, the DNA is packed within the nucleus as chromosomes. And in the prokaryote, supercoiled structure is present as a cytoplasm, within the cytoplasm as nucleoid. Nucleoid. See here, it's spread as a supercoiled structure in the form of nucleoid. No nucleus is there. Right in the cytoplasm, it is spread. Next, we go on to the physiochemical properties of DNA. There are mainly three properties. Solubility. The DNA is a polar molecule, meaning to say it is soluble in water due to its phosphodiester bond, that is sugar phosphate backbone. It is a polar molecule, hence it is soluble in water, and it is insoluble in salt and alcohol. Okay, that's why when you put alcohol in it, it becomes visible. The DNA becomes visible at the end of DNA isolation experiments. Absorption. Absorption says that in spectrophotometer, if you provide 260 nm, that is nanometer of UV, the DNA is absorbing, DNA bases can absorb the UV light. How? The free nucleotides, for free nucleotides, the light absorption is maximum, like 1.60 units. For single-stranded DNA, the light absorption is 1.37 units. And for double-stranded DNA, the light absorption is even lesser, that is 1 unit only. So maximum free nucleotides. Then next, single-stranded DNA has lesser light absorption, followed by double-stranded DNA, even lesser light absorption. Next, going on to denaturation and renaturation, based on which the PCR is built. That is polymerase chain reaction. The PCR machine, which makes a Xerox copy of the DNAs, multiple copies of the DNA, just like the Xerox machine. So, based on this principle only, the PCR is made. How? Heating will denature it, cooling will renature it, and for heating, higher energy is required if high concentration of cytosine and guanine is present within the DNA. Because remember, cytosine and guanine are having triple bonds in between, triple hydrogen bond in between. To make three bonds, more energy is required. Then breaking up two bonds in case of adenine and thymine, lesser energy is required. Lesser heat energy is required to denature adenine and thymine rich DNA than cgDNA, that is cytosine guanine DNA. Okay. The last topic of this session, that is the COT curve and COT value, that indicates the DNA complexity. It gives us an idea about the complexity of the DNA, and we get to know various information on finding out the COT value and the COT curve. See in this graph, E. coli DNA. See, this is the COT value on the x-axis. E. coli DNA, for example. For simplicity's sake, let us take it 10 to the power 3 for E. coli. Another DNA is there, that is calf DNA. This pink line, calf DNA, say for simplicity's sake, it's touching 10 to the power 5. So obviously 10 to the power 3 is lesser, 10 to the power 5 is more. So the calf DNA value is more. And if the COT or the COT value is more, that means what? The sample DNA sequence is unique, less repetitive. What do I mean by repetitive? That means suppose A, T, G, C as such, if it occurs a number of times within the long stretch of DNA, that means the repetitive sequence is more. And if it occurs very less number of times, that means the repetitive sequence is lesser. Unique sequence is more. See here, the blue part represents a single copy of DNA. That means unique DNA. That means it has having, the calf DNA is having a long region of unique DNA. Okay, so the COT value is more here. Whereas the COT value, this depends upon the individual DNA, like the calf DNA. Okay, whereas if the COT value is lesser, that says, that indicates that large number of repetitive sequence. Less repetitive sequence for more COT value and large repetitive sequence for less COT value. I hope this is clear. See here, single copy DNA means unique DNA. It's more in the case of calf DNA. So the COT value is more, approximately 10 to the power 5. Whereas the brown portion indicates repetitive intermediate DNA and the small pink portion represents sequence DNA, that is the non-repetitive DNA. I mean the repetitive DNA, the repetitive DNA, this part. So as the single copy means the unique DNA is more, so the COT value is more for calf DNA. COT value more in case of unique DNA, COT value less in case of repetitive sequence. So remember this. And now a short recap about DNA, deoxyribonucleic acid. DNA is a macromolecule with a double helix structure, that is twisted ladder, consisting of nucleotides, which in turn is made up of pentose sugar, nitrogenous bases, N bases and phosphates. N bases, that is nitrogenous bases, within the DNA consists of A, adenine, T, thymine, G, guanine and C, cytosine, such that amount of A, adenine equal to amount of T, thymine and amount of G, guanine equal to amount of C, cytosine. Amount of purine, that is amount of purine consisting of adenine and guanine is equal to amount of pyrimidine consisting of cytosine and thymine. Again, DNA plus histone means nucleosome. Nucleosomes form chromatin. Chromatin is condensed into chromosomes of nucleus, that is about DNA packaging in eukaryotes. In prokaryotes, DNA is present in the form of supercoiled structure in the cytoplasm of bacteria as nucleoid. Genes are what? DNA segments coding for an amino acid, that is precursor of protein, from which the proteins are generated, while alleles are variants of a gene. Variants of a gene on homologous chromosomes, that we have 46 chromosomes, that is 23 pairs of chromosomes, isn't it? That is 23 pairs of chromosomes, homologous chromosomes. Again, about the physiochemical structure of DNA, that includes solubility, absorption, renaturation and denaturation, based on which PCR is made. PCR machine is like a Xerox copy, making multiple copies of DNA. And then we studied about COT curve, and got values, which explains the complexity of DNA. Accordingly, we have the values. Thank you, dear students. I hope the concepts are clear. I hope you have got some information about DNA, genetics, the related terms and everything. This video, I think, is useful for you. Thank you for watching right till the end. Please do like, share, subscribe the video and do share with your friends, obviously. Thank you for watching right till the end. Thank you so much.